and then we have our annual mini symposium, usually in October or November. Today, we are very fortunate to have with us today Dr. Kniel from the University of Delaware. Next slide, please, Callie. As many of you know, it's been a very good partnership with a number of different organizations at all levels of government and non-government. So I think it's been a very fruitful collaboration and partnership in the agri-food safety in mainly focusing on the Ontario area, but of course covering off Canada and globally as well. Next slide, please, Callie. Okay, so just a little bit about Callie. And Callie is a professor in the Department of Animal and Food Science at the University of Delaware. Her research includes understanding the mechanisms of environmental persistence by bacteria, protozoa, and viruses in pre-harvest agriculture environments, along with studying mitigation technologies for these organisms in fresh and frozen produce. Callie is currently director of the Center for Environmental and Wastewater EPI Research. Her teaching is very broad and includes many different courses in the food safety area. Dr. Kniel is also co-author on the textbook Food Microbiology and Introduction and has more than 100 scientific peer-reviewed publications. She is very active in a number of food safety organizations such as IAFT and ASM, and of course IAFP, which is very dear to our hearts, where she was elected to the board in 2015 and served excellently as IAFP President in 2020. Next slide, please, Kelly. Just a few housekeeping announcements. Can you please turn the sound off of your cell phone? We'll be using actually the chat box for questions. So please don't use a Q&A, just use the chat box. And Susan Sabatini will be coming on later to help me with any questions that come up in the chat box. Also, the session is being recorded. So if anyone wants a copy, you can email Susan at the address that you see there. Many of us know Susan very well, and she does an excellent job coordinating along with Sumitra all of our sessions. So thanks very much, Sumitra and Susan, for the great job that you both are doing. Next slide, please, Kelly. So again, a big thank you to all of our sponsors and collaborators over the year. We could not function, that's okay, we could not function without the tremendous help and collaboration from all these various people that you see here on this slide. So without further ado, I invite you, Callie, to start off in terms of giving us sort of an overview of foodborne viruses and their global impact. So thanks very much.
Great. Thank you so much, Jeff. It's such an honor to be here. Please do let me know if something goes awry and you cannot hear me, or if something happens with the slides. So, but this is really quite an honor to be here with you all today. And I am sitting just sort of southeast of you, I think a little bit here, centered in between New York City, pretty close to Philadelphia, just a couple hours north of Washington, D.C. And this image here on the far right is actually the building that I'm sitting in. This is our College of Agriculture and Natural Resources at the University of Delaware. We have a 350-acre laboratory, as we call it, our farm right here outside our door. So it's a lovely place, and I welcome you, any of you that might be in the area, please, stop by and visit. So today I'm going to talk to you a little bit about GEMRA, and I'll tell you a little bit about what that is, and these amazing people here that are on this slide. And we're going to talk a little bit about some of these viruses that are shown here as well. So on the slide here from left to right, we have Yves, Kang, who represent the Food and Agriculture Organization of the UN. And then to the right of them, Moez, Juliana, and Akio, who are in the World Health Organization. And these individuals represent the two secretariats of those organizations that bring together multiple experts across many different areas related to microbiological risk assessment, and really do a wonderful job of wrangling all of us. As you know, faculty and scientists have a lot of different ideas, and so they really have a hard job in getting us to stay on task and stay on focus, and also illuminating areas that really need to be further explored. And so the specific area we're going to talk about today are these viruses. And so I had the pleasure of participating in two of these. I chaired the first part, which was related to microbiological risk assessment of viruses and looking at food attribution, analytical methods, and indicators. And then I also was able to participate in the one that looked at prevention and intervention measures. The executive summaries are both available for this, and I have some information on those at the end. And what I'm going to do is kind of take you through the information that's on those executive summaries, and then I suggest if you're interested more in reading the reports when they come out. The first report is on its way out, but still going through all the chains of approval. And then at the end, we'll talk more about, what's missing, and I'd love to hear your questions as well. And I think one of the biggest points, and I'll just kind of start off with it right now, even though it's a big need and a data gap, but it's one of the best things that we can do in terms of these global discussions is really talk about the need and desire for capacity building when it comes to virus testing, virus analytics, virology knowledge across the world. Because when we think of many parts of Africa or South America, parts of Asia, we may not have that expertise as much as we need, right? And so we've really worked well, I think, in North America in terms of building capacity in these sciences, but we need to continue to do that. So let's talk a little bit about GEMRA itself. So in response to actually a request of the 22nd session of Codex Alimentarius Commission in 1997, the Food and Agriculture Organization of the UN collaborated with the WHO to convene this series of expert consultations related to multiple areas in microbiological aspects of food safety. So GEMRA really came to be then in 2000. And each time they meet, they may cover different topics that have to do with aspects of food safety where there may be specific questions, or they may be addressing a very specific point from Codex, where there is a need for additional scientific information, or perhaps a need to update some of the language that might be there in documents. It's very important, I think, again, on a global platform that GEMRA exists because many countries are not as fortunate as the US and Canada to have the excellent federal organizations that they have, as well as many universities and things like that, and industry that all work together, I think, across North America, but they don't have that everywhere. So GEMRA really aims to develop and optimize utility of microbiological risk assessment as a tool to inform policy, to inform decision-making across the board for food safety. And we know as scientists how important risk analysis and scientifically based risk assessment can be. And it's this really big piece of risk management and risk communication. All of this really has to come together. Okay, did I lose you all? No, good. Okay, so sorry, my screen just changed. So when we look at the objectives of GEMRA, we're really thinking about assessing risks associated with foodborne pathogens, providing guidance on data generation and access to relevant data. Again, this is not something that might be available across the globe. And then developing guidelines on how to assess various risks, providing guidance on the application of risk assessment, and developing risk assessment tools. For example, this dose response curve that we see on the right-hand side. So GEMRA works by issuing calls. And so if any of you are interested, right, I certainly recommend that you look on their website for various calls that are coming out. And please, submit your information to that. It's a really amazing way to participate at this global level and to really meet amazing experts across the world. So it's such an honor to be able to be working with individuals that are there. It's a very transparent process, so you can go online and see all those different calls. They do have all the reports that are available. While the meeting is going on over a 5-day period, or even I participated in one that was on fruits and vegetables that was virtual during the COVID-19 pandemic. So that took a little bit longer, not just a week, but we must come together as a committee and affirm a executive summary before we leave. And then that becomes publicly available pretty soon after the meeting. So you'll be able to read that. And then there are a variety of technical reports that are available. So let's talk a little bit about the risk management aspect, just as a reminder, right? So when we think about risk messaging, I'm guessing that's something that many of you here are involved with. We're thinking about that interactive exchange of information to help communicate potential hazards to people, communities, thinking about risk assessors, risk managers, media, maybe interest groups, and the public, of course. Our direction really is to build trust, right? And to help people make better decisions about risk. This, I think, is increasingly difficult today. Following the COVID-19 pandemic, we do know that when it comes to truth and facts, right? Scientists are not always the people, the level that people go to. They're looking more on social media. Maybe they're talking to their friends. And so we really want to think about how risk management plays this role in risk assessment and risk communication. And it's becoming, I think, more and more important today, especially when we have this 24/7 information all the time, right? And so in one specific area related to today's topic is this third bullet, right? Using microbiological risk assessment in food safety management is an area that is still developing. And I think an example of that is what we know about norovirus transmission, right? When we think about exclusion of food service workers, right? We understand aspects of shedding, we understand aspects of norovirus transmission, but communicating that as a risk management decision is an area that is very difficult, right? That deals with trust and communication within a workforce. It has to do with whether or not we have time off for illness, right? There's so many social sciences, behavioral sciences, all of those things that come together. So I think that's one area where we're still really working on this, right? And we do want microbial risk assessment to be a useful decision tool, right? We want to be able to see that being used more frequently. And so it's really up to us as scientists, as communicators, to be able to get that information out appropriately. And so GEMRA does work in that. You can see that they've met, specifically, they have reports here on Vibrio as well as Listeria. They work in identifying various hazards, such as the viruses we're going to talk about today, and then breaking that down into characterization, assessment of exposures, and then characterizing those risks. And then trying to do that across the globe, right? And that, again, elevates this degree of difficulty, really trying to come up with statements that are both specific yet vague so that they can encompass all the needs of individuals. And so we really end up talking about differences between countries, often differences between high income, low income, and middle income countries. And I think it's important that we're trying to recognize all of that. So that's a bit on GEMRA. So now let's delve into a little bit more about foodborne viruses. There are many questions that we have about foodborne viruses. For example, which ones are the most important? Which ones should we be talking about in terms of risk assessment and risk management strategies? Does that vary by commodity? Are there global differences? And when it comes to trying to identify those questions or answers through surveillance, that's really tricky. We don't have enough of those answers really, because surveillance and data on testing and analysis, those are all fairly limited when we look at global questions. And so we'll try to go through kind of a framework of what we might do and how we wrote these different executive summaries and information that will be further elaborated on in the reports when thinking about identifying the viruses as the hazards, right? Characterizing what happens to those. How do we test them, right? How do you assess exposure? Are there specific commodities that are more at risk than others? And then what's this variability and uncertainty of the information? So as a reminder, this is a document that a postdoc who was working with me during the COVID-19 pandemic, Dr. Anderson Coughlin, and I came together with this because we thought it was really important to remind individuals that when we talk about foodborne viruses, those are very different from SARS-CoV-2 virus or even influenza virus that are enveloped viruses. So when we think about those differences, it's always, I think to students, it's a bit counterintuitive, right? You think you have an envelope, oh, it's got an extra layer, that must mean it make it more strong. But actually that envelope, which is phospholipids, is really important for the entry into the host cell, but it actually makes it more susceptible to things like hand sanitizers and cleaners. Whereas that really strong capsid that's like a puzzle that's being put together that norovirus and hepatitis A have makes it able to last on surfaces and be transmitted through surface exposure. Whereas SARS-CoV-2 and influenza viruses are not able to do that. And so this kind of an infographic that we came up to really help differentiate those differences in the viruses, I think kind of shows this pretty well. And this was important because food safety specialists are really used to thinking about how, indestructible, right? How these viruses are so strong and can survive on surfaces. And then we met SARS-CoV-2, and that is not the same, does not have the same transmission. So when we think about norovirus and Hep A, we're really thinking about those strong viruses. So during the first GEMRA meeting, the WHO and the United Nations came together to create this really nice video. So it's about 3 minutes long, so I thought we would take a few minutes and show this. It will.
In 2012, Codex developed guidelines on the application of general principles of food hygiene to control viruses in food, informed by GEMRA's scientific advice. But times have changed, and new research calls for updated guidelines. GEMRA is a group of international experts convened by the Food and Agriculture Organization and the World Health Organization to review the state of science on a particular topic, and to support the update of the Codex's standards on the control of viruses in food. GEMRA held a meeting in September 2023 to analyze the most recent research on foodborne viruses. Viruses are the smallest amongst the different types of microorganisms, and they are quite unique in that, unlike bacteria, they cannot multiply without a host, but they can persist in the environment and be transmitted to people through food.
Illnesses caused by foodborne viruses are of highest public health concern, and GEMRA has identified 3 specific viruses that require special attention. Norovirus is estimated to cause 125 million cases of foodborne illness per year, from which 35,000 are fatal. It is a highly infectious virus, and even foods with low levels of contamination can lead to outbreaks. These viruses are found mostly in prepared food, frozen berries, and shellfish, although any foods can become contaminated through infected food handlers. Hepatitis A is estimated to cause 14 million cases of foodborne illness and 28,000 deaths each year. These viruses are usually also transmitted through shellfish, frozen berries, and prepared foods. It is important to note that there are significant regional differences in foods responsible for illness for both norovirus and hepatitis A virus. The primary source of norovirus and hepatitis A virus is through humans. Foods become contaminated through contact with wastewater, unhygienic practices, or fertilizers that become contaminated through sewage.
A third virus, in addition to those previously mentioned, has emerged as a food safety threat. The hepatitis E virus infects an estimated 20 million people yearly, leading to around 44,000 hepatitis E-related deaths. However, there is yet no global estimation of what percentage of these infections are foodborne. In contrast to norovirus and hepatitis A, the main sources of hepatitis E are swine and wildlife, and the disease is transmitted by eating inadequately cooked pork and game meat. Foods that test positive for these viruses can be removed from the food system, but unfortunately, detection methods have their limitations and challenges. Better testing and surveillance could reduce viral foodborne illnesses. But also, preventing all viruses from even entering the food system is of paramount importance.
JAMR will provide additional scientific advice on effective control measures across the food system to prevent viral foodborne diseases.
So it was a pleasure to be asked to participate in that, and that is available on YouTube. I think they do a really nice job of explaining that and kind of give you a hint at the 3 viruses that we're going to be focusing on for the next few minutes. We met in September of 2023 in the first part, and this is where we were asked to come prepared after reviewing literature, looking at various surveillance databases. We also had presentations from individuals from regions around the world talking about their surveillance and detection of various viruses. And then we participated in an expert elicitation to rank viruses and the commodities that we thought and ended up agreeing on in terms of those of highest public health concern. And so we'll get at those pairs, but I think you just got a hint at those. And then we also discussed virus testing, also monitoring strategies, and then the relevance of viral indicators, which is a really important topic. And I will give you a quick look ahead that really isn't a wonderful response actually for viral indicators. So we have a lot more to discuss there. So we're considering both frequency and severity. The ranking of the viruses fell into these 3 groups. The first, as you saw in that video, is norovirus, followed by hepatitis A and hepatitis E, ranked in that order, and then a variety of different viruses that are transmitted through food, but we don't have a lot of information on them, and also the amount of these viruses that is transmitted through food is lower than that might be transmitted through drinking water, which was out of the purview of our discussions. And also, we don't have a lot of data on what might be transmitted through food. And so that includes rotavirus, sapovirus, enterovirus, astrovirus, and enteric adenovirus as well. So now let's look at those commodity virus pairs. As highlighted in the video, norovirus first being more of a burden associated with prepared foods. We had an interesting discussion in our group about that term, prepared foods. So I'm not sure if that's something that you use very frequently, or do you use the term ready to eat food? There are some differences in those in terms of where the preparation might be. And so that was an interesting discussion. When we think about prepared foods, we might be thinking about restaurants, or we might be thinking about quick service stations. There are multiple ways to consider that. Second, but with regional differences, are frozen berries and then also shellfish, right? So if we are looking in North America and in Europe, those frozen berries are really important when we think about attribution and virus illness that's associated with them. But if we were to travel to China, for example, we would not see the frozen berries there. We would see shellfish being more important. So there are those regional differences. For hepatitis A virus, you can see that a little asterisk there. So regional differences really across the board. And these are all as number 1, shellfish, frozen berries, and prepared foods being equal. And then for hepatitis E virus, and this was a big difference because previously, when these expert committees met to discuss these viruses, hepatitis E virus was not identified, and this was in 2008 and before then. And so this is something new that we're seeing is higher numbers, more importance in being placed on awareness and detection of Hep E. And so this is, I think, a really big statement from this group to include Hep E here. So we see pork most associated in terms of the public health burden, and then wild game following that. So let's look a little more now in depth at some of these virus commodity pairs. Shellfish, I'm guessing many of you are quite aware of transmission of both norovirus and hepatitis A virus through shellfish, right? The risks of these virus is being ingested with shellfish is greater, because often we are considering consumption of shellfish or bivalve mollusks raw, either or maybe only slightly cooked, maybe undercooked. Steaming, for example, we don't know all the time that those processes are validated to be able to inactivate viruses. I know this time of year, many people want to lightly grill these on the, on their backyard grills or at the beach. And so those may not inactivate viruses that can be inside the mussel, right? So mollusks are filter feeding organisms, and they're able to actually concentrate viruses that might be in the water. So we do have quite a bit of data on viral contamination of shellfish, really from around the world. This is one area that people continue to study and continue to get information on. Unfortunately, not everyone uses the same analytical tests, right? And so there's always some questions there, but we are trying to get this really good picture. And I think many people around the world do have this epidemiological data. And so that's very useful. The next is fresh and frozen berries. So as you all are very well acquainted with, I think in Canada and down into North America and the US, we've had numerous outbreaks around the world that have occurred, especially when we think about European countries, US and Canada, with fresh strawberries and raspberries, and then also frozen raspberries, strawberries, and berry blends. This is the company that was involved in a 2022 fresh strawberry outbreak across North America. These strawberries were grown in Baja, California and Mexico, and then they were frozen, and we had an outbreak again in 2023 with the frozen strawberries. And so I know here in the US and also in Canada, government, academics, industry scientists are working very closely to try to understand this contamination, try to alleviate contamination, and also looking at testing, recalls, all of those types of things. So a lot of research going on here. Just as a little bit of a research highlight, this is Alyssa Kelly, who was an undergraduate in my lab who looked at the ability of these viruses to maintain their infectivity while they're frozen. And so this is, again, why we see outbreaks that may be more extensive associated with frozen berries, and also berries that are grown around the globe and then frozen and then shipped into North America, where we are eating more frozen berries than anyone else up and down North America. And so we can see here on hepatitis A on the left, and then Tulane virus, which is a calicivirus that we use as our norovirus surrogate that can infect cells in the lab. So these are both infectivity assays. So we're looking at the TCID50, which is the tissue culture infectious dose that can make about 50% of those cells sick. And you can see, similar to a plaque assay, I should say, across 2 years of looking at these frozen berries, you can see that there is basically no change in the infectivity for hepatitis A, and also very little change in Tulane virus. A little bit of wiggle there on the raspberries. I think that's because as raspberries are frozen and you thaw them to look at them or to do the infectivity, they're sort of falling apart. So you might have some inhibition there in terms of infectivity, but the strawberries are maintaining that. And so when we think about these very large outbreaks that have occurred with frozen berries, such as those noted here on the slide, we're looking at the origin for those, and it's likely water, contaminated water, when these are very large. But when we think about frozen berries that might be contaminated by handlers or producers or harvesters that then get pooled and mixed and blended, we have, perhaps we need to go back to our good agricultural practices there and education. So the next food on the list were the prepared foods. There are ready-to-eat foods, and we do know for certain that norovirus is really important when we think about restaurant transmission, when we think about what's going on in food service. And so this one is perhaps least surprising to everyone. I thought I would just share this because I love these graphs, and you may not have seen these. So these come from Norostat in the United States, where they're looking at specific reporting of norovirus in multiple states. The gray areas here in both on the left and the right are the highest and lowest number of norovirus outbreaks that were reported between 2012 and 2020. And of course, we know norovirus is seasonal, right? The winter vomiting disease. So we'll see more in the winter months. The green dotted line is from 2018 to 2019 there on the left, and then 2020 to 2021 on the right. On the left, if you look at that solid red line, as we enter 2020, right? So we go from 2019 into 2020, you can see in April, it falls down, right? While the world is shutting down and people are not eating at restaurants, people are not leaving their houses for the most part early on. And then in the right-hand side, that red line goes from 2021 to 2022. So I think what is an interesting question when looking at these graphs is that role of restaurants, prepared foods, food service workers. Was it because we were eating less in restaurants for a while, and then of course we were getting prepared foods? Or is this really just a factor of less detection, less surveillance, because everyone in public health had transferred over to working on SARS-CoV-2 instead of norovirus? So kind of an interesting question there. So the next virus to look at is hepatitis E virus. So hepatitis E is quite unique among the foodborne viruses in that it's a zoonotic pathogen. It may have asymptomatic animal reservoirs, in specifically swine. So swine do not get sick. So you may have very healthy animals that may be harboring hepatitis E virus. In particular, the livers seem to be those when tested that where you have the virus there. So when we think about products where transmission of hepatitis E is more likely to occur, it's really those liver-containing products like sausages, also blood or other tissue or organ meats, for example. But Hep E is a very interesting virus, right? There are multiple genotypes. And so when we think about what's happening around the globe, those different genotypes factor into that. So genotypes 3 and 4 originate from infected animals and seem to be the major cases of foodborne hepatitis E. We've seen an increasing trend in those numbers in the last few years in some countries, notably in the US and Europe. These genotypes do cause acute hepatitis, which can occur in individuals, but can occur as more severe illness in individuals that might have underlying health conditions. They cause chronic hepatitis that can lead to cirrhosis and liver damage in people who are immunocompromised. They can also be associated with neurological sequelae. So undercooked pig products that include liver or raw sausage containing liver or blood, as well as liver pate, are the main foods that really are potentially at risk for contamination by Hep E. So this table here looks at the different genotypes of hepatitis E virus. First thing I want you to look at here is what's highlighted in kind of red there, the transmission route, right? So genotype 3 and 4 are the ones that we see that are more foodborne transmission. And if we were to now look at the geographic location, you can see that when we look at those waterborne cases of genotype 1 and 2, they're happening in Africa and in Asia and some of North America there in Mexico, but really genotypes 3 and 4 are happening in Asia and also in developed countries like in Canada, the US, and Europe. So we have very different viruses here, different genotypes. And so when we think about testing, we have to figure how does that all come into play? So it's not necessarily very easy to answer. So the other viruses that were discussed here by this group include those that are ranked in this order: the rotavirus, saprovirus, enterovirus, astrovirus, and enteric adenovirus. So these are various viruses that cause diarrhea or gastroenteritis in humans. Astrovirus, for example, you might think of more likely to occur with children. Rotavirus, because there is a vaccine that's being used. We still see quite high numbers of that, but then associated with food, right? But maybe as that vaccine becomes more widely available, we'll see if those numbers go down. We did talk about emerging viruses, right? So you're probably familiar with Nipah virus, an enveloped henipavirus, which is transmitted through contact with infected animals. It can have very high mortality. It's something where we need to continue to think about how animals are being housed around fruit trees and where we have the reservoir of the fruit bat that might be there. So we've seen very large outbreaks in Malaysia and Singapore, part of Indo-Bangladesh outbreaks associated with consumption of raw date palm sap. So we have to think about messaging, risk messaging there as well. Of course, avian influenza virus, H5N1, right now we have in the US in dairy cattle. And of course, thinking about individuals and the messaging there, not consuming unpasteurized milk, because we know the pasteurization process can actually inactivate viruses that might be there. And then the coronaviruses, right? We know we've had MERS transmitted in association with dromedary camels. And then we know that the unlikelihood of SARS-CoV-2 being transmitted through food, but noticing or keeping those in mind as our emerging viruses. So we also talked quite a bit about detection methods, right? There are validated detection methods that exist for norovirus and hepatitis A. We have international standards, the ISO methods, and then we also have the Food and Drug Administration in the US with the BAM methods, that's the Bacteriological Analytical Manual methods. The FDA BAM are more specific in terms of the different kits that have to be used, the different ingredients or companies or framework for processing, whereas ISO's a little bit more lenient in terms of which systems can be used to accomplish detection. They're both validated for very specific matrices, but not all matrices, right? So there are still limitations in terms of RNA detection across the board in terms of how those are both using qPCR techniques, and what now do we think about with digital that's coming on board in more and more laboratories, which is an absolute quantification instead of comparing it to a standard curve. But how does that different type of detection come into play with both those methods? So there are quite a few challenges that still exist, different matrices. What about environmental detection systems? What sequencing should be done for confirmation? And how do we implement these technologies and these standardized methods into low resource countries? It's particularly difficult if you want to think about that with the FDA ban, because that relies on a ultracentrifugation, which many labs don't have anymore, those types of machines. So we also talked about indicators, right? And asked the question, can indicators be used for viral contamination? We are seeing a lot of work in this in environmental waters and with shellfish. But still the question exists, what's the appropriate viral indicator for foodborne contamination? And bacteria are being analyzed, bacteriophage are being analyzed, plant viruses, animal viruses. Which one is the best, right? We still don't have a solid answer for that for all conditions. For example, we might look at pepper mild mottle virus, which is shed by humans at very high levels. This is one that you can actually follow in wastewater. Crassphage is also ubiquitous in humans, shed by healthy humans. Bacteroides seems to have a lot of merit as well. It's very common in the human microbiome, but we may see regional differences with Bacteroides. So I have two examples that kind of touch on this. So this is work that was done by Kalmia in my lab, who's a PhD student. And so here we were monitoring SARS-CoV-2 presence in wastewater in the state of Delaware and also regionally on campus here at University of Delaware. And so here you can see some really nice correlation between the presence of SARS-CoV-2 virus and also pepper mild mottle virus. This was from 2020 into 2023. But now let's throw norovirus into the mix. And you can see we do have some positive correlation there between those three viruses, but norovirus is pretty different, right? With that big spike, of course, during the winter months. And so interesting there. So it's not perfect, but it is usually present. So now I'll transfer into the second meeting and talk briefly about what the objectives were for that. And so this was a more specific area, really looking at taking those virus commodity pairs that we've just talked about and then looking at developments that have occurred in terms of control of foodborne viruses since this last report was published, which was in 2008. And our consensus really is that prevention remains the cornerstone of control of foodborne viruses because viruses are environmentally persistent and resistant to many treatments. So we really need to work on prevention. So now we'll go back, we'll look at some of those. So what are we thinking about with shellfish, right? Well, we know that they can concentrate viruses. So how do we get viruses from not entering their waters, right? Or can we use those indicators as an indication of fecal pollution, right? Are there sanitary surveys that should be used? Better information on when there are point source or non-point source pollution events, septic tank failures. These are things we see in the US with older infrastructure. We really have to keep on top of these things. Also, can we use colophage as an assistant when it comes to depuration? We know that there are some issues with depuration. It's not a perfect process, but if you add colophage to that, it seems to be able to help. Also looking at tertiary wastewater treatment, more of that being done if possible. But what happens now when we have significant rainfall events, as we're seeing more and more frequently, and we have runoff and sewage issues? So that might exacerbate the problem. In terms of fresh and frozen produce, again, we're looking at contamination either by sewage sludge or water sources that are fecally contaminated by humans, or are we looking specifically at handlers? So we want to be able to have good communication, right, about those risks and good risk messaging, and going back to our good agricultural practices, good management practices, good hygiene practices, right? Maybe treatments for irrigation water, maybe more discussion about personal hygiene. Also having plans to divert contaminated produce to go into jams and jellies or juice production. Now the prepared and ready to eat foods, really the information there needs to focus on exclusion of infected food handlers, right? Exclusion from the workforce. And that can be difficult depending on, again, regionally, good communication, all of that's really important. So if we can't always rely on that, what can we do in terms of gloving, surface disinfection, or hand washing? So I urge you to look at these reports. I know they're going to have some wonderful tables with lots of data if you're interested in that. Kind of fun to pore over those and look at differences there. Food handler exclusion mandates do not exist globally. We don't have compliance with hand washing practices globally. We don't have formalized guidelines for cleanup. And this is one thing we talked about, and something that I think is also a good takeaway. There's a difference in terms of surface cleaning and disinfection after an emetic event or an infection event, or where we might have defecation or something like that, versus normal cleaning and sanitizing. And so we want to be able to identify those differences. Now, when it comes to Hep B, right, we're thinking about that pork and wild game meat, that zoonotic transmission. So it's sort of a different way of thinking. How can we reduce illness from consumption and use of these raw or undercooked meats and tissues, right? Avoid infection or by reducing contact with animals or contact with untreated pig manures. So some of those aspects may really include beefing up biosecurity, which is good. That can help us with some of those emerging viruses as well. Also, understanding inactivation strategies for hepatitis E, right? So 20-minute treatment at 70-72C for a pate-like product can inactivate Hep B. That's a lot more than it would take to inactivate just the virus, which is about 2 minutes at that temperature. So we need to know those numbers. So we need more research there. So there are many data gaps, right? We need better detection around the world. That needs to work within our risk management framework. Also, there's a difference between detecting viral RNA fragments versus an intact virus. Can we get to that point? Can we understand those differences? Can we get to a point where we have standardized methods? We need that global capacity building for training, adoption of methods, surveillance, right? We need a lot more information when it comes to viruses, especially in specific regions of the world. So there are many needs. that are being outlined in these two reports. Assessing mitigation strategies. Of course, a need is the limitation of virus cultivation, right? For norovirus, we're often using these surrogates like the Tulane virus that I mentioned, right? We can't do everything in organoids or the enteroids, which is a wonderful system for norovirus, but it's very costly and still has pretty extreme limitations in terms of using it for environmental testing. So what else do we have? Can we turn to wastewater surveillance or remote sensing imagery, right? Let's turn to some of the use of drones, some of these hydrographic dye studies that might predict virus dispersion, right? We have a lot of great technologies that are available. How can we learn from those? What about these indicators? Let's go back to those and try to see how they predict occurrence, right? That means we need more surveillance. Validation of viral surrogates for relevant viruses and testing applications, looking at those mitigation strategies. I think many of you online may be working in that area. Surface disinfectant and hand sanitizer formulations need to continue to be tested, so we have ones that work against norovirus and hepatitis A virus. There are vaccinations. For example, there's vaccine for Hep B, but it's not implemented globally or available. And so, and the Hep A vaccination also has limited use globally. So how do we have those conversations? Are they worth having? And then region-specific risk assessment, right? Determining the value of specific interventions. So lots of needs and lots of room for all of us to be involved in that. I note these slides will be available to you through the recording. Both of these QR codes should lead you directly to the executive summaries that were mentioned. And I really, have to acknowledge the amazing people who are in the Secretariat for FAO and WHO and all of the outstanding GEMRA colleagues that worked on these two reports. So thank you for the opportunity. Thank you for your attention today.
Thank you very much, Callie, really for, in my mind, a really brilliant overview of the GEMRA and Codex work, foodborne viruses. You've covered off all of the major ones that we are all concerned about and really their global impact. So I think we'll now open it up to question. And I see Susan has made it. So thanks, Susan, for coming on. And you can help me with the questions and going forward. Thanks.
So thanks again for a great presentation. We're getting lots of feedback about the excellent presentation in our chat box. Before I forget, the recording will be available. I have put my email address in the chat box, so if you would like a copy of the recording, please email me. We've also gotten a question with regards to the actual slide deck and whether that will be available. So, okay, I will speak to Callie about her slide deck later, and then again, if you're interested in it, please email me directly, and we'll get that out to you. Okay, so first question we have is. Great presentation, very informative. So the question is on the challenges using phages as indicators, especially CRA SS phages. Unlike colophages, we can't isolate most of them in the lab, and we still don't have conserved genes that can detect and question. I hope I relayed that well.
Yes, yes, absolutely, Susan. Yes. No, that's a great question. And I think there's more and more studies being done on cross-phage. The Center for Produce Safety has funded some work in their detection on fresh produce. And so I think if you want more information, you can look at their website and you can pull up reports there. That's Center for Produce Safety. We do have, I'm happy to share the primers that we use for that. But you're exactly right. There are limitations, right? And we will even find surveillance of fresh produce or of environmental samples where we find norovirus and we don't find any indicators, right? And so does that mean it's just a viral fragment because the RNA is very stable, or what does that mean? So I think, yes, you're acknowledging those, there are limitations, right, about testing, how we can all test for them and their usefulness too.
Okay, Susan, I'll take the next one there. So, Callie, so with the prevalence of norovirus in frozen fruits, what are some of the ways that consumers, what are some of the things they can do actually to protect themselves, ensure it's safe? Because we can't, for the most part, cook them or whatever.
Yes. So this is a tricky one, right? Because we do see, I mean, contamination is fairly low, right? There have been large outbreaks, but if we look across the board, contamination is fairly low. So the risk associated with that is also low. I do have colleagues that are concerned, and especially if cooking or making smoothies for children or older adults or people you think that might be more immunocompromised, you can cook the berries and then refreeze them. It doesn't, I don't think it changes the quality too much. I mean, it will change your consistency, but you'll still get that berry taste. So if you wanted to use it in a smoothie or something like that, I think that's an alternative. Not perfect, but luckily we do see lower risk. And I know that I have colleagues that are working in this industry. They are really, adamant about trying to reduce the risk. And so working very closely with a lot of berry growers and frozen food production environments.
Great. Okay, another one Callie came up with, obviously, food safety testing is known to be limited. What risk management strategies are there for decontaminating frozen fruit?
Yeah, so I think that there are, again, those industries are looking at different technologies. Can they implement things like different surface sanitizers or wash water treatments or filters for irrigation water? And again, working with the handlers for touching, but also maybe there's some UV treatments or things like that that can also be used. So I think that there's room for testing and for adaptation of different technologies that are available.
Great, Susan, you want to take the next one there? Okay.
Yep, this one's about Hep E. So they were concerned about hearing about the rise in Hep E as a source of infection. So some health units sometimes ignore testing foods once norovirus is identified in stool. Should the messaging change?
Oh, great question. Yes, I think that's what this executive summary and report will say globally, that, yeah, Hep B should be on our radar a little bit more. And maybe we don't call it an emerging virus anymore. It's there, right? And we need to recognize it. In particular with, foods like raw blood sausages. These are culturally important foods, liver, pâtés, things that are enjoyed by individuals. And so we want to be cautious in that messaging. But yeah, I think so. I think we should be talking more about viruses, that they're there, and how do we test for them appropriately? How do we communicate those risks associated with specific products? Yep. I think it's an excellent point.
Callie, just as a follow-up to that, I know that there's at least one country that puts out information to the public consumers to actually, if you're in a high-risk group, let's say if you have liver disease or something, to avoid things like undercooked pork or pate. What are your thoughts in general? Do you think we need to do more in terms of risk communication to the public? Because of course, I don't think any of them maybe even heard of hepatitis E.
No, that's wonderful to hear that people are doing that. I think that's really important. I mean, I know in the US, our healthcare system is a bit messy. And so we very rarely have those good conversations with your physician. And so if there are public health groups that can do that can get information out to especially groups of individuals that are at risk, I think that's wonderful. I'd love to see more of that, good risk messaging, not alarmist in nature,
Right.
of course, but just really good messaging. I think that's such a critical piece today.
Yeah, that's a follow-up, I guess, is a lot of people ask, how effective is washing berries and vegetables to reduce the risk to people who consume them?
So from what I've seen, it can help to reduce some contamination. probably most familiar with people talking about how, the bacteria may be stuck. And so same for viruses, you may not be able to remove them completely. Again, what we're looking at though is really low level contamination events. And so again, it might be really hard to remove that completely. I think if people are concerned, for example, I know I have family members who have been on chemotherapy, and during that time, they did not want to take a risk and get norovirus. So really cooking everything, was important to them. I think the best news that we have is that most contamination that occurs is at really low risk or low levels. And so we're not seeing, thousands or cases. And most of the time for norovirus, we're able to resolve those with our immune system.
Kelly, one other thing, what is your thoughts in terms of, there are a lot of public health officials who call for mandatory Hep A vaccination for food handlers. We've had a lot of discussion about this. What are your thoughts in general on that?
I think that's a great question. I actually asked my students this question in one of my classes. And I think it's tricky. I do think that it's a great vaccine. It has really low risk of any effects, of negative effects. I ask everyone in my lab to get the vaccine since we work with it. I think it's a great alternative. And honestly, I do think that it's very helpful to companies, because if they can vaccinate their workers, they wouldn't have to worry about outbreaks and then all of the economic repercussions that happen. And it's a fairly affordable vaccine. So I know I'm going out on a limb saying I think that people should be vaccinated and that companies should do that. But I think in the end, it could really protect people and, protect individuals who are more at risk. And we see older populations now that are more at risk. We see some interesting aspects of maybe, globally, people who are more at risk of Hep A, even people who were vaccinated as younger, or now everyone's getting older. And there's some interesting aspects in terms of the immune protection. And yeah, I think if we have this as a tool, I think it's good to use.
A question came up about food irradiation as an option for viral inactivation for raw berries and shellfish.
So I can't remember offhand if it's approved here and what the amounts would be, but you do get some inactivation. So I think cold plasma, I think that chlorine dioxide, those two show a lot of promise when it comes to inactivation of viruses on produce, maybe on shellfish as well, but certainly, yeah, there's room for more studies if consumers are going to accept those types of technologies. I think the chlorine dioxide and cold plasma, or excuse me, like the activated plasma, which the electrons bombarding the food, I think there's a lot of potential for those that consumers may be more willing to ingest those. Also, high pressure processing works to inactivate viruses on shellfish, but I think that that's sort of phasing out from what I understand in the industry, not using it as much as maybe they had hoped to at one point.
And yeah, in Canada, it hasn't, to my knowledge, those hasn't been a submission for raw berries or shellfish for radiation. So from what I remember, let's see, maybe I'll do maybe one last question, Kelly, in terms, I don't want to put you on the spot here, but okay, if, there's been so much discussion about doing a recall when you find an RT-qPCR positive on a frozen berry, and what are the real public health implications when you know that you may not have viability? Just to get to your general thoughts on that before we close.
Yes, so this is one of my favorite subjects these days. We're doing a lot of work in our lab looking at what we find in the environment when we find Hep A, and what can we sequence, what can we learn from that. I think one of the big takeaways from COVID is the COVID outbreak or pandemic is that we know that viral RNA can be very persistent in the environment, right? When we were detecting particles, right, in the nasopharynx forever, we stopped doing PCR tests because we knew that it wasn't an infection. So I think we can translate that same takeaway into this situation and try to understand more about levels of viral detection, pieces and fragments. What are some better methods for that? And yeah, my lab is actually trying to piece some of those things together by looking at persistence in the environment, detection by digital PCR, sequencing. They're not easy answers, but I think it's really important because we have to balance detection of virus with food waste and with economic issues and all of that together.
Great, thanks. All right, so again, I wanted to thank you, Callie, for a tremendous talk. I think I can see by the number of questions, a lot of interest here, a lot of people on the call. And Susan, maybe I'll hand it over to you. You want to just close us off here, or?
Yeah, just thanks again for a fantastic seminar. Really appreciate all the great information and good questions as always. So thanks everyone for joining us at our seminar. We're going to take a break for the summer, be back in September, and we will have our annual symposium in November. If you have any questions about today's seminar, if you want the recording, slides, etc., please email me and I will follow up with you. So I just want to wish everyone a great summer. Have a good break and when you get your holidays. And yeah, we'll see you back in the fall. And thanks again to our speaker, Callie, for today. Thanks, everyone.
Thanks, everyone.
Have a great day.
You too. Bye-bye.
Bye, thank
Bye-bye.
you.